Welcome, everybody. Thank you for joining us for the colloquium today. Before we begin, just like to first take a moment to acknowledge that UBC's Point Grey campus, where we are hosting this colloquium, is situated on the traditional, ancestral, and unceded territory of the Musqueam. And also point out that in the BC Lower Mainland, we live and work on the unceded territories of the Squamish, Tsleil-Waututh, and other Coast Salish peoples. So it's my great pleasure to welcome Dr. Samantha Anderson today, who joins us from Arizona State University. She did her graduate training at Notre Dame, where she got her master's in clinical psychology and PhD in quantitative psychology. Her methodological work focuses on important but often overlooked design elements of psychological studies, particularly regarding study design and things like sample size planning and power analysis. She's done a lot of work also on meta-science topics. So, for example, as she'll be talking a lot about today, looking at methodology pertaining to replication studies. Personally, I've always been very impressed with her work, and particularly how well it's simultaneously very rigorous and sophisticated from a methodological and statistical standpoint, while also still being very practical and immediately useful to researchers across areas. I think she's a good exemplar of someone who kind of bridges, the gap between practice and methods in a way where both sides of that are supported very well. That said, I know I'm not the only one who has this opinion. She obviously gets published a lot in all the top journals. She's developed a lot of popular tools that are used by a lot of researchers, for instance, for sample size planning, and also has gotten a lot of awards both from major psychology organizations like APA and APS, but also quant-specific ones like SMEP, where she was recently awarded the Early Career Researcher Award. So yeah, I'm excited to hear more about her work, and I assume we all are as well. So please join me in welcoming Samantha Anderson.
Thank you all so much for having me and for inviting me. It was 105 degrees Fahrenheit in Phoenix when I left, and this is my ideal weather. I like a light rain and like 50s to 60s. I have to say Fahrenheit here. I also tried to use the Canadian date way of doing things. So I'm really happy to be here. As Jason mentioned, some of my quantitative work involves meta-science topics. I study replication from a really broad perspective, and just trying to find ways to improve the design and rigor of our studies so that they are more replicable and research literatures are more interpretable. So we're going to talk about some of those things today. There it goes. So my interest in this topic started way back when I was in grad school, and we had our very first statistics midterm. And I emailed the teacher and asked if he still had the midterm from 2013. And this was the question. And everybody in the class got this question wrong. So we were given a chance to work through it and think through this question. And it was my first experience of just having my mind blown with relation to this topic. So I wanted to, I think I have to go a little closer to here for this to work. I wanted to walk us through this problem to get started. And so with the question, we were given just a little power analysis table. Since we all got it wrong, I guess it wasn't clear to us how we would use this table in our results. And so we start out, there we go. So we start out with a hypothetical research literature where studies fall into two camps. It's very simplified. We either have studies that are looking at truly large effects, so think Cohen's large standardized mean difference, or the study is testing a truly small effect. And they only fall into those two camps. And like a lot of psychology, in this fictional literature, most of the studies are studying small effects. So 90% of the studies have a small effect in reality, and 10% have a large effect. And we sort of simplified things. We said, okay, there's only going to be one statistical test. P less than 0.05 is going to get you published. And the studies are using a sample size of 20 participants per group. And the first part of this question asked us to think about what the result would be in the published literature. So not the hypothetical literature of all researchers conducting these studies, but in what's going to get out into the published record that researchers are going to be able to read. And it asked how many studies or what proportion in the published literature would reflect a large effect. And of course, we know that very few of the studies being conducted actually have large effects. So in a perfect world, we would expect that only 10% of the published studies would be large effects. I knew ahead of time that that wasn't going to be exactly true. It was just the magnitude of the difference that changed my mind a little bit. So it turns out that what we were looking for in this problem was just the probability that we reject the null hypothesis, given that the effect is large. And to solve this, we just need to use a Bayes' theorem type problem, which says that the probability of event A given that B has occurred is not going to be the same as the probability of event B given that event A has occurred. And it turns out that we had all the information we needed to sort of solve for this probability in the population. And that's where that power analysis table came in use, because one of the quantities that we need to solve this is essentially the power or the likelihood of getting a statistical significant result when the effect is small and when it's large. So we had that information. We also had in this very simple literature some prior probabilities for the effect sizes of 0.9 and 0.1. And so when we solve this, we see that 45% of the studies in the published literature reflect large effects. So of course, that's quite different than the 10% that were conducted in reality. So we have a literature that's not really reflecting reality. And then the second part of this question was the one that I think people got wrong. And it ended up being a little bit of a trick question because it asked, okay, most of the studies are testing small effects. So how often will we see a small effect size reported in one of these published studies, right? So if the study is a small effect, an accurate representation of that would be if the published study reports a small effect size. So it asks how often are we going to see those small reported effect sizes in the literature? We can actually solve for the minimum value of the standardized mean difference that we would see in this literature. So the smallest effect size in these studies using only 20 participants per group. And when we solve for that, we get a minimum Cohen's d of 0.64, which is larger than a medium effect size. So the reason this was a trick question is because in this fictional example, we will never see a small effect size reported in the published literature. And of course, this is a hypothetical example, but a lot of it did reflect reality. And this was, like I said, my first brain explosion going on, where I saw how different the published literature was and how inaccurate a reflection it was of what the studies were actually being conducted. And so I'm interested in the bias that we saw in the published literature, so how the effect sizes that were reported were generally much larger than we would be seeing in all of the studies. And I'm also interested in the fact that the balance of the small and large effects wasn't kept up in the published literature. So we have an incorrect estimate of the heterogeneity across these effect sizes as well. And so if I put on my glasses here, I think I look a little bit like this cartoon. But what I've been trying to do since that exam question is do anything I can to learn more about how we understand research literatures and how to improve the accuracy of our literatures. So what I'm going to talk about today is a little bit about replication in general, and I promise this will kind of connect into replication. But I'm going to talk about 3 different studies that focused on different aspects of replication and research literatures. So first, I'm going to talk a little bit about planning replication studies. So this is what we think of when we think replication. We're going to do a replication study. Then I'm going to talk about questionable research practices and some of their impacts on the research literature, and a little bit about heterogeneity as well. And at the end, I'll talk very briefly about some thoughts I've been having about the future of replication and what we can do. So let's start out with the definition of replication. And it's essentially obtaining the same results or conclusions in a new sample. So from one study, confirming those in a new study with a new sample. And if you've heard about replication, it might have come from some of these headlines. Psychology was said to be amidst a replication crisis. Everybody freaked out about it. Apparently, it spread to other disciplines like a contagion. But it got me really interested in thinking about replication a little bit more broadly. And so rather than only thinking about a single replication study and how we can kind of design that, I see replication as a process that sort of begins with our original studies. So how can we design studies to make them more replicable? How can we think about avoiding questionable research practices, thinking about how publication bias and our way of publishing affects our results that go into the literature, and thinking about designing those studies to avoid some of these issues with replication. Of course, the way most people think about replication is this middle box. This is where we're designing a study specifically to try to reproduce or replicate the effect that we saw in another. And we can think about special ways to design those studies. And then finally, I like to think about replication really broadly because all of our studies are in some way pulling from earlier research. And so what we end up with are these really big research literatures with a lot of different studies. And we're trying to sort through these literatures to motivate our new studies, to figure out what the past evidence says. And there is a lot of heterogeneity and inconsistency in these literatures that makes it really hard for us to figure out, like, what's the consensus on this particular effect that we're looking into. And so one of the topics that's going to come up a couple times is heterogeneity. And I want to be clear that I think heterogeneity is a good thing on its own. So we don't expect our effects to generalize to different contexts and settings. In fact, in psychology, this is one of the most interesting things when we do find that there are differences. But what we'd like to do is be able to attribute when two effect sizes are different, we can say, oh, that's because of this very clear difference between these studies. In reality, though, there are so many reasons that make studies different from each other that it really becomes hard to pinpoint what is the cause of the heterogeneity across studies. And so to try to help out, I like to divide heterogeneity into a couple categories. Conceptual heterogeneity, this is the good stuff. This is, oh, I'm using a different population, or it's an urban versus a rural context, or I'm doing this in two different countries. These are those results that we expect will have variability. But the problem is we often mix in methodological variability as well. Some of this is rather unavoidable. We don't have unlimited resources, and so sampling error is always going to be a little bit of an issue. We can't measure constructs perfectly in psychology, so that's an issue. But I'd like to point out that I think some of this variability might be avoidable. I'm going to show how some of it depends on using questionable research practices. So if we can kind of diminish that, we might be able to reduce some of this extra variability so that we can really see the true variability and heterogeneity kind of shine through. So to begin, I'm going to start with a study that's more focused directly on replication. And this relates to my work on sample size planning. So replication studies tend to use an estimate of the effect size that comes from the study that they're trying to replicate, comes from the original study. Some of my previous work showed that this was problematic because, as we saw in that first example, published effect sizes tend to be much, much higher than the true effect size. And your statistical power actually depends on the true effect size, not the effect size that's reported. So I developed some software that adjusts effect sizes for both publication bias and uncertainty before we use that in sample size planning. And we thought this was, great to use for replication studies because there's often a prior estimate that probably has those publication bias and uncertainty issues that researchers can use. So just to show you how much of a problem publication bias and uncertainty can be, Suppose we read a study in the literature that reports a Cohen's d of about 0.6. So this is a little bit larger than a medium effect size. And what we can see from the red curve on the graph there is that the likelihood of a particular population effect size that's coming from, it's really wide. That red curve is really wide. So it's saying with this small sample size of 25 per group, that study in the literature doesn't really tell us that much about what the likely population value of the effect size is going to be. And we can also see that the red curve is centered at a value much lower than 0.6. That spot where the curve peaks is sort of the maximum likelihood, the most likely value for the true effect size. And using a method developed by Hedges, I calculated that the most likely effect size producing that 0.6 is only 0.16. So the problem comes when you try to plan the power for your study and you think the effect size is 0.6, you don't collect enough participants because the effect size is actually much smaller. But then if this study used a much larger sample size of 150 per group, we can see that the blue curve is not only much narrower, but it's also centered. I picked an example where it's centered around the right value. It won't always be that case, but just to give an idea of how this sample size planning works, we don't have to worry about the top column or the top row. That's the non-centrality parameter. It's a little bit more directly related to power than the effect size, but we can use the second row and take a look at the effect size. So I simulated a population of studies that had this F squared effect size of 0.014. And then I looked at 3 different methods of sample size planning. So in the first one, we just directly take the effect size that we see reported in a single study. So to do that, I drew one published study out of that literature. And that published study got a much bigger effect size of around 0.08. And so if the researcher just uses that effect size directly, they get a sample size suggestion from like G-power or something like that. But unfortunately, because the true effect size is a lot smaller, their sample size just isn't big enough and they don't get very high power. There's a method that corrects for uncertainty that helps a little bit. It brings that effect size a little bit closer to the true value. But in this case, in my simulation, I was also looking at publication bias. And so the third method corrects for publication bias as well. And I did cherry pick one where it was right around 80% power. But in general, when we remove the publication bias and uncertainty from these effect sizes, we can get more accurate sample sizes for our desired power. And I realized that 500 plus participants might not sound that great to have to go and collect, but at least you know how many you would need. So one of the really most important projects that led us to thinking about replication was this reproducibility project Psychology. I don't want to criticize this project too much. It was really fundamental in expanding our thinking about replication. And the researchers worked really hard to try to carefully replicate 100 different results from psychology journals. And this, in the past, the low replication rates had been criticized in relation to statistical power. And I had thought that it was just what I showed you, that maybe a lot of these researchers were using effect sizes that were too big. But I looked a little bit more carefully at the critiques of power, and they were more focusing on the fact that they only had one replication study of each original study rather than like the many labs projects that have multiple replications. And so I thought it would be interesting to go back, and if I'm going to be selling this technique for replication, to see how people were calculating their sample sizes in these replication studies, and how that compared to what they could do if they corrected for publication bias. So we saw that the researchers were using well-intentioned sample size planning. So a lot of the studies were using larger sample sizes than the original studies, so that's moving in the right direction. However, we noticed that about 2/3 use the effect size directly from the original study with no corrections to it. And 26%, about a quarter, didn't use any effect size information in their sample size planning. And so we asked a couple questions. First, we wanted to know how different the sample sizes that the researchers actually used differed from the sample sizes that our sample size planning program would suggest. And so we saw that if we were only correcting for publication bias, there was about a 25% difference. So they were using sample sizes that were about 25% too small, according to that. If we also adjusted for some uncertainty, that number goes up to about 50% there. We also asked how many of these replication studies would have been approved for to go ahead based on their sample size if it was a requirement to correct for publication bias and or uncertainty. If we only required publication bias correction, only about 15% would be approved. And if we required some uncertainty correction, only about 5% would be approved. And if you wanted an accurate confidence interval, that was even more difficult to achieve with these studies. We also looked at a couple of the predictors that related to the eventual p-value that was obtained in the replication study. And one of the things we saw was that how different the sample size was compared to what this sample size method suggested resulted in p-values that were less significant. And also surprising effects. So effects that researchers deemed to be surprising were less likely to be replicated from sort of the p-value perspective there. And I want to take this not as a negative. At this time, replication was really, really new, and people didn't necessarily have all the tools to use different sample size planning. But this is sort of a reminder that it's not necessarily the case that all of these unreplicated studies are, were lying in the first place, or were false positives, or used QRPs. Part of it can just be a sample size issue with the replication study. I'm working with the replication project Health Behavior, which is going on right now. It's a very similar project, but I was a little disappointed that they're still not requiring publication bias or uncertainty corrections. They're suggesting that you use a smaller effect size than reported, but they're not really specifying a specific way to do that. But we're trying to move in the right direction with these things. So now I'd like to broaden things a little bit and think more about the research literature and how some of the decisions that other researchers may have made in their studies can affect the literatures that we have to go through as researchers. So let's talk about questionable research practices. I generally like to say that these are still common. I will say no one in this room has ever done any of these questionable things. And one of the things we know is that when you do some of these maybe sketchy things, maybe we didn't know for a while that these things were sketchy, we can often have higher false positive rates, higher type 1 error rates than we would have had. And there's a reason that I'm still wanting to study QRPs, even though, especially when I'm speaking to quantitative researchers, they say everybody knows we're not supposed to do those anymore. So the reason is we do have some solutions. So for pre-registration and registered reports, a lot of the increase in transparency helps with this. But I think another route is to give researchers a little bit more nuanced information about the effects of their QRPs. So instead of just false positive rates, let's think about how they affect other aspects of the research literature. There's also been a sense of studies asking, how does this QRP affect my study, my type 1 error rate, something like that. I'm going to think a little bit more about the whole research literature. And then even if QRPs are declining, and more recent studies seem to show that that may be the case, the existing research record is still full of studies that were conducted earlier in the before times. And we don't know when we're reading a study, whether it might have been subject to this. So they're still really affecting the whole literature that we have access to. So I'm going to start out by talking about selective reporting. So this is the type of study where you want to know if a certain color of jelly bean causes acne. And you just test each color of jelly beans one by one by one. If you test enough colors, you're going to eventually get something that's statistically significant. And then you write up your study saying that green jelly beans lead to acne. And I'm going to differentiate between two types of selective reporting. The first one, and I'm using the terms coined by this article in Business Psychology. The first one is cherry picking, and despite the name cherry picking always being used in a negative sense, this one is the more benign of the two, and it's very, very common. So a lot of times researchers will have a grant, they'll collect lots of data, and they have a bunch of different measures that purport to test the same construct. We can argue whether or not these three questionnaires really all test depression in the same way. Those of you that study measurement probably know the answer is no. But researchers think, these are all studying depression, and they might choose the one that has the smallest p-value, thinking, maybe this is picking up on the construct a little bit better. Question trolling is the one that's a little bit worse and a little bit more sinister, where you don't really have a hypothesis. You have a bunch of different constructs, and you sort of pick and choose. You fish through your results until you find something that's statistically significant. So I'm going to look at both of those. And I wanted to know what the consequences of these are in a research literature where some researchers are engaging in these things. So I wanted to try to be realistic and think, we're not in this awful literature where all of these studies have these problems, but how might these things affect the broader literature? We also looked at different study and field level factors that could influence this. And the one I'd like to point out is publication bias. So imagine we just dropped p-values completely. Someone argued to me that researchers who would have been p-hacking with p-values could still kind of do the same thing with effect sizes. So you might pick the CESD depression questionnaire because the effect size of that one is the largest. So we wanted to see, okay, if we dropped p-values completely, but people still maybe picked the larger effect sizes, how would this affect the research literature? And rather than just looking at error rates, we wanted to look at mainly bias and heterogeneity. I won't worry too much about the details of the simulation, but we had about a third of researchers not do any selective reporting, a third selected among 3 variables, and then a third selected among 5 variables. This let us compare amongst those and then look at the overall literature with all of these researchers together. We varied things like the sample size, the effect size, the correlation among the dependent variables, and the presence or absence of publication bias. In terms of bias, what we wanted to do is separate out the publication bias. This is what would happen anyway, even if there was no selective reporting. When we submit to journals, we're more likely to submit and we're more likely to get accepted if our p-value for our focal effect is less than 0.05. So we're going to have publication bias. But on top of that, do we have additional bias due to selective reporting, and how much is that? And then the other outcome was heterogeneity. So if studies appear more similar to each other than they really are, that can make a finding appear more robust than it really is. So that's not great. If they appear more different than they really are, that can make it just more confusing to figure out what's going on with these different studies. So neither of these things are good. We wanted to compare whether the heterogeneity was increasing due to selective reporting or decreasing due to selective reporting. In general, we found that selective reporting does add bias above and beyond publication bias. For cherry picking, this was about a 15% increase in relative bias. For question trolling, it was quite a bit higher. That's the worst one of the two. And in the scenarios where we lowered publication bias, either by having larger sample sizes or through ignoring p-values completely, the proportion of the bias that was due to selective reporting actually increased. And I'll show you that in just a second. So here is one of those busy graphs with a lot on it. But the bars are showing the bias. So if we had an accurate reflection of the effect size, we shouldn't see any bars at all. The light blue is publication bias, and the dark blue is the extra bias added from the selective reporting existing in the literature. And the top row is in the regular literature where we kind of need a significant result to get published. And in the bottom, we can see that there's no publication bias This is where researchers are just sort of tweaking the effect sizes a little bit. So what we can see in these figures is that there is dark blue there. So cherry picking is leading to some bias above and beyond publication bias. We can also see that the bias worsens for question trolling. So this graph is showing question trolling. The darker blue parts are higher there. And if we focus a little bit more on the bottom panel where we don't have publication bias, we can see that the amount of dark blue there is much higher than in the top. So that shows that getting rid of p-values by itself isn't going to solve things. We have to motivate researchers not to do the same, essentially do the same thing with effect sizes. I will say, though, that the overall height of the bars goes down, so reducing publication bias is a good thing overall. We want less bias in general. In terms of heterogeneity, the results weren't completely consistent. This is what makes it more interesting. Sometimes the heterogeneity was higher than it should have been, and sometimes it was lower than it should have been. This is partly due to a couple things. So researchers in this fictional literature were making different choices. I know I talked to some of you today who are doing multiverse analysis. We know that making different choices with regards to our analysis can kind of create a garden of forking paths that makes things appear more different. And then we have publication bias, which is going to serve to truncate the distribution of effect sizes. And then we'll see selective reporting works in a couple different ways on heterogeneity. So to show you what that looks like, so this graph is just showing a distribution of all the effect sizes that we would see in the published literature. And in the very first row of the table, that is the literature where there's like no publication bias, there's no selective reporting. If we could see every study, we would get this nice normal distribution of effect sizes centered right around this population value of 0.5. And what we do in the rows below it is we add in publication bias, we add in selective reporting, or we add in both. And we can generally see that the distribution truncates as we add publication bias, and it kind of shrinks a little bit and shifts a little bit upward when we add in the selective reporting. So generally for larger effect sizes, the selective reporting reduced heterogeneity compared to what it should be. So it made studies look more similar than they really were. On the other hand, with a smaller effect size, we can have what we call type S errors. These are results that are published in the opposite from expected direction. So the true effect is positive. Group A is better than group B, but we get a significant effect that says group B is better than group A. Having more of those serves to increase the heterogeneity across the effect sizes in the literature. Very briefly, we then extended this to optional stopping. I don't plan to just go through every QRP, we're gonna stop. But optional stopping is where you collect some data, you stop, you check your p-value. If you don't like it, you collect a little bit more data, and you kind of go on like that until you collected your maximum sample size. And so we wanted to ask generally the same questions. We looked at some meta-analytic results as well for optional stopping, and some different types of optional stopping. So we looked at different timings of optional stopping, as well as how many times you check the data throughout. We also tried to really vary a lot of things to make it even more realistic. So we varied the sample sizes in the literature rather than just having them all be the same and things like that. And so thankfully my student is great at making flowcharts. So this was one of our simulations. We had half the researchers engaging in optional stopping based on a study by John and colleagues in 2012. And those researchers varied in terms of how many times they checked the data, as well as whether they checked the data early or towards the end of data collection. And to keep it fairly simple, the results were quite similar in terms of bias. The optional stopping was adding, you know, a fairly large amount of bias on top of publication bias. And in terms of heterogeneity, optional stopping tended to increase the heterogeneity. And what it tended to do is sort of pull up effect sizes at the, I should do it backwards, at the higher end of the distribution. So it kind of expands the distribution a little bit, which increases heterogeneity. What's interesting is that if you're then conducting a meta-analysis and you calculate the I-squared metric, the I-squared will actually go down despite the fact that there's more heterogeneity. And that's because we have the same amount of true heterogeneity. We're just increasing the amount of total heterogeneity relative to that. A reviewer on that study mentioned, publication bias is a little bit more complicated. It's not that all significant studies that are 0.05 or less get through and no non-significant studies get through. So I took that to heart, and I wanted to think about a little bit more nuanced ways of thinking of publication bias. So I have two additional types here. This middle one is lower publication bias. What that curve says is like, your chances of getting published go up as your p-value gets smaller, but maybe some slightly non-significant ones are kind of getting through in this literature. We sort of softened our insistence on p less than 0.05. And then the high one on the far side there is even more stringent. So it really wants you to get a small p-value to get published. So I just wanted to see if looking at selective reporting in these different literatures, if it just had additive effects or if there was any sort of interaction in how QRPs would affect the literature with different types of publication bias. So I looked at questions relating to the original study literature, which is the same as what I've done before. So how are things affecting bias and things like that. I also looked at meta-analyses, so what happens in this literature if now we want to conduct a meta-analysis of these studies. And then I looked at how it would affect planning replication studies based on this literature. And just showing some preliminary results right now. In terms of bias, it's fairly straightforward. Publication bias, as it gets stronger or worse, it adds more bias. Selective reporting adds bias. They're kind of additive effects. Slightly interesting in terms of heterogeneity. So in a perfect world, with just sampling error, we would expect a heterogeneity estimate of about 0.22 for these studies. The ones I show in green there, those are too big. The heterogeneity estimate is larger than it should be. And the red ones are too small. So the ideal, the most accurate situation is if we can get as close to 0.22 as possible. And in the case here where we have no publication bias, the best we can do is no selective reporting. That definitely makes sense. No publication bias, no selective reporting, good. But the best we can do in the case of publication bias was actually in the high selective reporting condition. So this is not a signal that we can go ahead and do selective reporting because it has so many other bad consequences. But what I was seeing was that the publication bias truncation and the way some of the selective reporting was increasing, they were kind of working against each other and a little bit canceling out the negative effects on heterogeneity. So I really want to look at other cases, other definitions of publication bias to look at these results a little bit more. What I'm just showing here is that the bias extends into meta-analysis. Of course, that's expected. And then in the graph on the right side, I'm showing that the bias will extend into issues with sample size planning for replication studies based on this literature when researchers are using selective reporting. And so the Buck sample size approach is shown here. And we can see that using that will reduce, will make an improvement, but it's not meant to do anything with respect to QRP specifically. So it can only help so much there. So just to summarize, we saw additional bias due to QRPs. We saw a little bit complicated effects for heterogeneity, and that these effects extended to replication studies and meta-analyses. All right, so let me check my time quickly. So we have till 1:50, right? Okay, great. So I was doing all of this stuff with QRPs, and then I realized, like, I don't know that, maybe it's just me, but I don't know that we all have a great idea on what we should really expect for how different two studies should look from each other in terms of their effect sizes. Like, let's say we have replication studies. So we do one original study, we try to replicate it, and we see that the effect sizes are different from each other. We know that they're, of course, always going to be different. It would be weird to do a study and get exactly the same effect size because we have sampling error. And that's one of the very first things that we learn in statistics class. We learn about sampling error and sampling distributions. And I realized I just wanted a more, maybe this was just more for myself, but a more practical way of saying, like, in ideal conditions with no QRPs, what could I expect about how different two effect sizes are going to be, if they really should be the same? And so that's what this very preliminary third study is about. We have many cases where we have access to multiple studies that are either replications or sort of tweaks, very similar studies on a phenomenon. So in social psychology, I know it's really common in some journals to have to submit like 5 or 6 studies at a time. And all of those studies go into one article, and they might be a little bit different from each other. One might be in the lab, one might be outside, some changes made from study to study. And so we often have access to like multiple effect sizes in the same article. With replications, of course, if we do multiple replications on an original phenomenon, we can sort of see how the effect size bounces around over time. And people have also suggested the idea of within study replication. So if you're going to do a study, maybe you should replicate yourself first and kind of submit both studies at the same time. And so with all of that, we've already talked about all the sources of heterogeneity that we deal with in psychology, those substantive differences that we have across studies, and then sampling error, measurement error, QRPs, and simply all of the different choices that we make in analyses. And so I wanted to simplify this quite a bit. So I'm just going to think about 2 studies. So we only have 2 studies right now, study 1 and study 2. And in the simplest case, we can think that study 1 and study 2 are testing the same effect. It really, like in a perfect world, that effect size would be exactly the same in study 1 and study 2. So I wanted to know, well, due to just sampling error, how different should the effect sizes be in study 1 and study 2? And then I also wanted to know what would happen if we made a tweak to the paradigm in study 2. So maybe we expect there to be a little bit of a difference. We expect, oh, we changed something in our setup. We want to see how that, affects the effect size. And my question is, like, can we tell that the tweak actually works? So if it turns out that the effect sizes are going to be really different, even when they should be the same, then it's going to be a lot harder to tell if the little tiny tweaks that we make to our studies actually result in a difference in effect sizes. So I started with some analytic work to look at the expected value for the difference in absolute values of the effect sizes of 2 studies. I'm using the standardized mean difference. The reason I'm looking at the absolute value is that I don't really care which study ends up with the larger effect size. Study 1 could be larger, study 2 could be larger. With sampling error, the sign of the effect is going to bounce around. And so I really just care about the magnitude of the difference. So if there's no difference in reality, on average, how big a difference will we see in the research literature with no QRPs? And to do this analytic work, I'm assuming that these are 2 independent studies from each other, 2 different samples, and that the true difference in effect sizes is 0. I'm not going to go through all this math, but I started with the sampling variance of the effect size for an individual study. And then calculating the standard deviation of the variant, or standard deviation of the difference in effect sizes. But that's not enough because we want the absolute value, not the true difference. So we can use what's called a half T distribution to calculate the expected value of the absolute value or the difference in absolute values. A half t distribution, if you know your student's t distribution, it's just that distribution folded on top of itself right at the middle so that it only has positive values. There's no negative values. It's not symmetric anymore. And in doing that, we can look at a couple different scenarios. So I'll just start with the worst one, just to make it interesting. So if both of these studies are using 20 participants per group, which in some areas is still a common sample size to use in psychology, the true difference between the effect sizes is 0, but we see on average a difference of almost a Cohen's medium effect size. So if you got one study, you're just reading it in the literature, and it has an effect size of close to 0. Now, maybe that won't get published anyway, but has a really tiny effect size. And then you pull a second study that seems like it's doing the same thing, and that study reports an effect size of almost 0.4, you would probably think these studies must be different. There must be something different. At least I would think there must be something different about these studies. You know, I got to figure out the reasons that they might be different. But this is literally the average or expectation of how different these studies should be, even just due to sampling error alone. So you can imagine if we add QRPs and all the other messiness, we are seeing a lot of heterogeneity in our effects that's hard to explain. Of course, it gets a little bit less exciting as we increase those sample sizes and reduce that sampling error. Just to make things a little bit more dramatic, I also looked at percentiles here. So in the worst case, this means that 10% of the time, if we're looking at 2 identical studies with 20 participants per group, 10% of the time we're going to pull 2 studies that are different by a magnitude of almost Cohen's large effect size. So even more, if you see a study with an effect size of around 0.8, and then you see another one that has an effect size close to 0, your first guess is probably, oh no, the effect is the same, it's just sampling error. At least mine wouldn't be. So this just helped me, I guess, really put into perspective how much, you know, sampling error alone can have on our results. So very briefly, I conducted a small simulation just to test this a little bit further. So just very simple independent samples t-test. And then in the simulation, I varied whether the tweak the researchers put works or doesn't work. So in some cases, they change something and it turns out that change has no effect on the true effect size, that it doesn't work. And in other cases, the tweak does have a small effect in sort of changing the effect size, something that you did in the study, the context matters. And then I just varied the sample size between, you know, rather small to a little bit larger values, and the population effect size of the 2 studies. The main points that I've seen so far is that there is a lot of overlap in the distributions of the difference in effect sizes when we compare the case where the tweak doesn't work and when it works. And so I think it'll be a little easier if I show that. So in these graphs here, we have 2 distributions, one in blue and one in pink. And in the blue distribution, the 2 effect sizes are the same in reality. And in the pink distribution, the tweak works, they're different. And we can imagine that we just kind of, you know, we don't have access to all of these studies. We just have the 2 that we've done. So we're just kind of pulling out one little piece of those distributions. And especially in the smaller sample sizes, we're often pulling something that's completely overlapping between the blue and the pink distributions. Meaning we pull out 2 studies and it will be impossible to tell whether they come from the world where there's no difference between them in reality, or they come from the world that the tweak that we made actually works in reality. And even with a fairly large sample size of 150 per group, we can see that there's still a lot of overlap in those distributions. So I'm a visual person, so this just helped me know, well, like replication is really, really hard because this is in the simplest case where we don't have any of these real world things added. And it can be really hard when we get replications that seem to contradict each other, to know what to do with that information. And this just here is showing it in a little bit of a different way. The expected value of that difference in effect sizes. We can see that a lot of that difference in effect sizes is just due to sampling error alone. And the tweak, whether it works or not works, that just adds a little bit to the expected value. But relative to what we already have from sampling error, it's not that much. So I'd like to expand this a little bit, maybe consider more than 2 studies, to consider 3 or more studies. But this was helpful to me just to see how much heterogeneity really affects us. So in general, it was difficult to decipher whether study 1 or study 2 came from the same population. And I think in general, it's difficult for us to interpret things amidst heterogeneity. I know the hardest part is when I'm starting a research paper and I want to motivate my study and say, previous studies found this. And then I find all these different things and I'm like, previous findings were mixed. I can probably find that sentence in a lot of your papers as well. So we're all just trying to like unmix those previous studies. So finally, what I'm hoping to do over the next, rest of my career is think about the overlap among replication, research design, or research practices and study design, and think about how all these factors interact in the real world. And to finish, this is just some thoughts that I've been having on consensus in replication. So we've had this replication crisis. It got psychology thinking, hey, not only should we look at our studies and question some of the rigor, but replication is important and we should start doing replication studies more. So we've seen these initiatives like the Many Labs project, the Many Babies projects, the RPP studies, and similar initiatives in other fields. We're doing a lot of replications, still not enough. I think we need more of this. But as we do these replications, we get a little bit more information here and there, right? Each new study adds something. And I've just been wondering, how do we eventually achieve some sort of consensus? It would be really easy if we did a bunch of replications and all of the effects started coming out pretty close to each other. And we could say, oh yeah, I feel like we understand this phenomenon. But in reality, with these replications, I was just talking to a researcher today who's doing some of those replications. In reality, the effect size is often going to be seemingly really different. And we have to think about what are all of the possible reasons why these effects are different. Is it just sampling error? Is it low power in the original study or the replication study? Is it QRPs? Is it measurement differences? Is it just the garden of forking paths and researchers making different decisions? Are there contextual differences? There are so many possibilities for this heterogeneity. So I'd like to sort of think through different criteria that we could have for consensus in multiple replication studies, and think about how those criteria might interact with different ways of defining replication. So we can take a p-value approach, we can take a Bayesian approach, we can look at the difference between effect sizes. There's lots of ways to evaluate whether a replication is successful or failure. But either way, all of those differences kind of interact with each other and may make it difficult for us to actually say something about these replication studies. So that's just kind of what I've been thinking about that I'd like to think through over the next couple years. But that is all I have for today. So I want to thank the collaborators on the projects that I've mentioned, Scott Maxwell at Notre Dame and Ken Kelly there, and then 2 of my graduate students who did a lot of work on this. And we should have some time for questions if anybody has those, but thank you.
All right, so yeah, just heads up for Q&A. I have these little dongles for recording. They're not amplified, but I'll do my best to pass them out.
Jess just pre-registered the question that she thinks I'm going to ask, so we'll see if she's right. But first, I just want to say thank you so much. That was, I think I just had the same experience that you had when you took that midterm, where I was like mind blown. So interesting. Thank you so much. I have a lot of thoughts, but one quick comment and then 2 questions. Sorry, that's a lot, but you can pick if you want to. More chances to win for Jess. So first, just a comment, like I've sometimes joked with my grad students that we should call it power guessing instead of power analysis, because power analysis gives you this feeling that there's a right answer, and it feels like that captures some of the uncertainty. And this is going to be really like guessing to this question and asking you to kind of go beyond the data. You probably can't really answer it, but like in your work, would you, based on your work, if you were trying to calculate an effect size, like are there sample sizes where you're like, I don't even want to include that study in my power analysis?
Yeah, so sometimes people will come to me, they'll say, I have these multiple, like, I'm not doing a replication, but these are 3 similar studies, and I don't know like which one to use. And I will say, I wish my method allowed, maybe that's something I should work on. I wish my method allowed for sort of a meta-analytic estimate of the effect size to then correct the publication bias with that. Because I think overall that would generally be stronger than using from one prior study. But sometimes they do come to me, and one of the studies, I sort of look at it, I say, that's 15 people. I literally know that the power analysis software isn't going to have enough to work with there because there is so much uncertainty in that situation. So I think in general, I'm really in favor of cumulative science, because that small study, doesn't have to stand alone. If we use meta-analysis or Bayesian methods to sort of build upon that, and it can eventually be more than 15 people contributing, that would be great. But yes, there are some cases where either I'll tell them to use the larger of the studies that they come in with, or we'll say this isn't the sample size planning method to use, because it really does require enough information about the prior effect size. Mm-hmm. And so there are other methods where you sort of come up with a minimally interesting effect size that you can use, and you don't need prior data for that. So sometimes that's when I'll recommend that, or like an accuracy sample size planning approach, because this is one way where the prior research like really does affect us when we're planning our new studies. We have to kind of rely on that. So
awesome, thank you. Okay, I'm going to just ask one more quick question, which is, you showed the D1 minus D2 difference, which was like so striking, but all up to like fairly small samples. Were those, if in an experiment, were those condition sizes or those were total ends?
Those were condition sizes.
Oh, okay. So not
the largest one would have been 300.
Okay, gotcha.
I really should, like, it's newer work, but I really should show, like, maybe I can show there's like distinct non-overlap at like 1,000 or something.
Right. And so I guess what I'm wondering is like, how big a sample do you need where you start to see like, okay, the D1 minus D2 actually matches up with our intuitions, where it's like there's a little there, but it's not much.
That would be great. Yeah, I haven't done those larger sample sizes yet, and I think that would be another way to kind of push the point home of like, ooh, we really got to do this cumulative science, because we know we can't all have these studies with 1,000 people per group, especially in between subjects research. So that might be another way to kind of show like, we would need ridiculously large sample sizes to have this like small amount of expected heterogeneity. Yeah.
Got it. Thank you. Hi, thanks. Was Samantha and I have been in a writing group for, I don't want to say how many years. Long enough. Okay. We were like new faculty back then. So it was awesome just to see you talk about your work. So thank you for that. So when you first started talking about publication bias, I was thinking about registered reports. And I was thinking about when you're setting the publication bias to 0 in a simulation, can we imagine that that's a reality where all journals only publish registered reports?
And that would be way different than what I did, because with the registered report, you wouldn't be able to like force the larger effect size out, right?
Oh, sure.
Yes.
But then actually it kept going, and I was thinking, no, is it some? But you can also think of registered reports as a way of changing QRPs.
Yes.
So I was just curious, in a simulation where you're trying to operationalize or like a mathematical model of what happens under the case of registered reports, how would you think about that? Would you think about that as a lever that pulls on QRPs? Would you think about it as a lever that pulls on publication bias? Both? What problems do you think they do and don't resolve, basically?
Yeah.
And you can maybe tell people a registered report, if you're not familiar with it, is a publication option where the data analysis plan has been peer reviewed. So it does not rest on what the results are. So you can imagine that publication bias is less of an issue. But yeah, I'm curious how you think about that in your simulation world of the literature.
Yeah. So in general, what I didn't do in the QRP simulation that I think would have made the results even more drastic is I varied the sample size to see overall how does that affect the bias. But the sample size that you're starting out using is going to affect your temptation to use the QRPs to begin with. So the people in the 20 group per group condition are probably more tempted to say like, oh, I probably don't have enough statistical power, right? I might have to resort to some of this p-hacking. And so the proportion of studies using QRPs would probably be higher for smaller studies than larger studies. And where I think that comes into play with registered reports is I think you're right that they pull on multiple levers at the same time. So if we can infuse more, and what I'm trying to think about is like the overall literature, right? So I think it would look different if I only looked at registered reports and said like, okay, if every study was a registered report, how would this change things? Versus thinking about like registered reports slowly funneling into the broader literature and now being, 5%, 10%, eventually all studies will eventually be registered reports. I do think, at least in my experience with registered reports, that they would not only lower the publication bias quite a bit, but also lower the temptation, right? Because it's more than pre-registration. So there's been some work with pre-registration that says it hasn't really solved the QRP problem. And that's because when you pre-register, while it's a really good thing, not all journals will really even read your pre-registration. People may not look at it. And so oftentimes there's a lot of changes that are made between the pre-registration and the eventual article. Whereas with a registered report, you're directly talking with the reviewers and they're directly reading your research plan before they approve your projects. So I think registered reports are one of the best things at reducing QRPs among themselves and then eventually reducing like the percentage of studies that have QRPs. So if I had the option to look at information from even two equal sample size studies, and one is a registered report and one is not, I would focus more on the registered report because I think that is more robust. But yeah, ideally, I'd like to take a more positive spin on things and not just be like, oh, everything's bad, but think about all the improvements that we're making and show how those can eventually make a big improvement on the overall literature that has all the studies. Good question.
Yeah, sorry, I was handed the mic and I wasn't sure if I was next.
Multiple mics so I can question.
Okay.
Awesome.
Just go at the same time.
Yeah,
Just whoever yells louder.
You gotta fight for it.
Yeah. Great. So thank you so much for this. Inside me are two scientists. One is someone who is a sucker for like measurement and great research practices, and the other scientist is developmental. So oftentimes I find myself at this crossroads of like, well, yes, I can see that I need 150 babies, but I would like to graduate, so that's not going to happen. So it's no surprise that I'm very heavily involved in many babies. So that's great. So my question is kind of two sides of the similar coin, because this issue I imagine is not unique to developmental. If you have difficult to collect participants or expensive to collect participants, or like there's only 3 guys with this one disorder that you're trying to study in the world, you can't achieve the level of power that maybe will let us sleep at night, right? And the second one is related to that, which is I think one thing that I see is fundamentally different about psychology compared to something like physics is that our culture is changing so quickly that what we're studying today might be very different. Like the people we're studying today might be very different from people 30 years from now. So how do you reconcile the need for cumulative knowledge, that is science, with the rapidly shifting landscape of like humanity, to put it really nicely?
Great question. So with the first one, I will say that I'm often in these conversations where I know I'm like, oh, my program is telling them they need 1,000, and I know they're doing MRI and they can, So that is one of the reasons I'm big into cumulative science generally, because I also like to tell people, I focus a lot on sample size, but it's still one part of your study. People will do these really large online studies with like MTurk, and oh, that's great. They get all these participants and they're like, oh, Samantha, you'll be so happy. But if they have completely forgotten about all the other validity aspects of their design and their measurements, now we have a study that's strong in sample size, but not in other areas. So it's really just one piece of everything. And I'm really sensitive to the fact that we have to work in the real world with practical limitations. I'm really in favor of like multi-site studies when we can do those. But then with your second question, I think that's a really good point, is that should knowledge always be cumulative? When should we bring in the previous stuff and when should we leave it behind? An example I like to use is attitudes toward women in the workplace in 1950 and today. If I do a replication study, I hope it's a failed replication. But I don't want to put those in a meta-analysis together and say, oh, these are the same things. So that's called effect size drift. It happens, across context, but also across time. And there's a paper called like, you can't step into the same river twice, right? So it's one of the reasons effect sizes are always different. And yeah, I think that's a really good question that probably depends on the context, but sometimes we don't want to include previous studies. Sometimes there's something that happens that fundamentally changes things. And then we have to build off from there. And that's a reason why I'm really still in favor of some like direct replications. People will say, oh, that's not really adding knowledge, or you should change something about the replication. But if we can do a couple replications in close sequential, like, then we don't have to worry about that effect size drift, and we could get a really good idea about the effect at that time. Again, all of these things require resources and time, and so, yeah, but that's a really good point, that things often change and we can't include everything. Yeah.
Hi, thank you so much. I think this was just so great. I echo what everyone else has said, so thank you. I think my question is a little bit about like, some of your examples made me think we should just throw out everything and just like change fields or something. But others made me think that like a lot of the stuff making me think the first thing might also be wrong, right? Like I was struck in the beginning about the size of expected heterogeneity that you would get even when the replication is still testing the same thing. Yeah. Even at like fairly large sample sizes, at least given my world, right? So yeah, does that mean that like we should have thrown out everything we used to do because that was all biased and bad? And we should also throw out all the replications because those were bad. And we should throw out the next thing we're doing because those are bad in these other ways. And like, I feel lost.
Yeah, I feel that way a little bit too. Yeah, maybe we should just throw everything out. Let's start fresh. You guys can all be conducting the first psychology studies, and you'll be in the intro psych textbooks. But yeah, like all those studies you learn in intro psych, I wonder about those. But I try not to have such a dark reflection about things. I once called it, instead of the replication crisis, I saw it more as a replication dilemma, because I think we are generally doing things really well in psychology. Because what we study is so difficult, we have to think a little bit harder about our methods and our analysis. I think we think a little harder about how we measure things. And I was reading something about how the speed of light, which I thought was just like a fixed value that everyone knew, has not only like has a confidence interval around it, but also has bounced around over time. Like the measurements are all over the place for the speed of light. So I don't even think physics has it figured out. I will say sometimes there are some studies that I read that I say, I just don't think this information is quite as useful. Maybe it was useful at the time, but maybe I'm just looking and they're using methods or QRPs that I can see, and really I'm like, I think maybe that one could be set aside. And so that's why when we're doing things like meta-analysis, I think we want to think about the quality of the study that we can tell. But I do think we've learned a lot, and that we're seeing that some of our effects really are holding up, and those ones that are really rigorous and robust that we keep seeing kind of gives me hope. And I also think that the heterogeneity can be interesting too, because it gives us somewhere to go and more questions to ask to sort of refine those questions. So I try to be optimistic and think that, you know, we've done a lot of good research in the past and we can kind of keep expanding from there. But yeah, it's a little bit hard not to, yeah, sort of think, let's throw everything out. But yeah, thank you for that.
I have kind of an off-the-wall question. As we know, like psychology is kind of unique in using standardized effect sizes, and most other fields use unstandardized effect sizes for various reasons. But one of kind of a new reason that I haven't thought about that occurred to me when you were talking about the tweak study is that I wonder if the standardized effect sizes also exacerbate the problem of those distributions not being separable because they add the variability due to the denominator in addition to the variability in the means. And I just wonder, like, if you've thought about it or if you know how much that makes it worse.
It very well could be. Yeah, I haven't looked at like unstandardized mean differences or different contexts altogether, like regression or whatnot. That would be really interesting to look at. Yeah, we do rely a lot on standardized effect sizes. There are some people that really don't like standardized effect sizes. I am generally, in a lot of situations, still in favor of them more than other people. But yes, like, and I did use the unbiased version because the bias in the standardized mean difference can also make things worse, worse too for really small sample sizes. But yeah, that's a really good point. I should play around with other effect sizes. It'd be nice to show some positive things too. So if I could show that unstandardized leads to more separation in those distributions, I mean, that would be great. It'd be great to have a more positive angle too. So that's a really good thought. Yeah.
Well, while I have 3 microphones, I might as well ask something. Yeah, thank you. Really enjoyed the talk. One kind of specific thing I'm curious about, you mentioned how I think it was like journals aren't, you know, forcing anyone to look very systematic in sample size planning using effect size, like systematically look at things like publication bias, et cetera, and correcting for that, but do have a recommendation of like using something smaller, like a smaller effect size than what you'd think. So I'm curious, like, yeah, just in general, what you, and yeah, there's lots of work on talking about like the smallest effect size of interest. So yeah, for one, I'm curious just like your overall thoughts on, you know, the broad utility of that in terms of sample size planning, whether it's for a replication study or just for a single study. And then second, like, how are there cases in which using a more systematic approach like you've done would lead to a very different, like, ultimate sample size or effect size estimate?
Yeah, I think it's hard because sample size planning, there's a lot of perspectives on it. So there's some researchers that see it as sort of, you want to be the most conservative you possibly can. And so you want to pick the very smallest thing that, you know, could be, so either the smallest effective interest or a lot of uncertainty correction if you're doing that. I try to see it as using the information we have to get the most accurate sample size that we can have with the information that we have. So that's a little bit different ways of looking at it. I will say I recommend different approaches in different situations. So I often recommend approaches that are not my own. I'm not just like selling my own approach. But I was reading, I often try to steer people towards the minimally interesting effect size. But I find that when I'm working with psychologists, that tends to be really difficult. If I'm working with health researchers, they often have an effect size where they say, like, this is the smallest amount of clinical change that we would even care to talk about this treatment. If it doesn't reduce blood pressure by this specific amount, it's not going to get approved, you know, by the FDA, or I don't know what you have in Canada, sir, Canada FDA. So it can be really difficult with the questions that we ask in psychology to come up with this objective method. In one study, they tried to do this. And so they had different experts come in and try to say what they thought the smallest effect size of interest would be for that particular paper. And their distribution of what people thought the smallest effect size would be was very large, wide distribution. And so the paper was sort of about how it was a little difficult to do that in practice. So yeah, I think there's no perfect method of sample size planning. but I like to see people using some information rather than just only using the same sample size that their lab always uses or using some sort of convention. I think some sort of customization usually will help us get closer to what we need than sort of just like winging it. But yeah.
Well, I think we're about at time. We got to clear out for the next classroom, but thank you all for coming. And thank you, Samantha.